وبركاته. والله تعالى في القرآن الكريم بعد أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقهوا قولي. صلى الله على سيدنا وحبيبنا محمد. Inshallah ta'ala, I'll be going our meals from from you and all of us. Ya Rabb al-Alameen. Ya Rabb. Ya Rabb.
What is this one? It looks super short, but it's like. Yeah. There's two.
I was probably starting on second order. Okay, no, I might be wrong. Four. Over. Like 50. I was wondering that. Yeah. Later on. I'm so mad at you because
It's hard.
Yeah. I just
Yeah.
Stapler, yes, I do. It will be on the lecture 7. No, I'm sorry.
Not this here.
This is not B, but if you look at the previous okay, maybe I'll talk about that now. I have a stapler as well. Just give me one more. Okay. Amro, do you mind if you can close the door? Okay, if it's not working, I don't mind if they're kept together. That should be okay. Okay, everyone. So we're about to begin. As I go through intros, I just want to highlight a few things. One is for a generation time assignment. Okay, So I understand some of you may have had outstanding questions. I likely answered them all, but if there were some outstanding questions, as well as those that were received extensions by Dr. Brent, please send me an email for that proof as well as the timeline, because I need to have confirmation of this as well. Now, to answer, okay. When you're officially done, I just want to highlight this because I think it's a bit of a concern. In your semi-log graphs, are you seeing a line that is curved, i.e. non-linear or linear? They're non-linear. Any other thoughts? Any other thoughts? On semi-log paper, are you seeing a linear, like a function that is linear or non-linear? This is essentially what you should be seeing. If you plotted your points correctly, you should see with the logarithms that they eventually either come close at specific time points, right? You went up by 15 increments for the microplate reader, and then 30 for the Bible counts. But the idea is that you should see a linear curve because on normal Cartesian paper, i.e. just regular paper, this would have been going logarithmically or even exponentially. So the idea is that you would see, yes, on regular paper, a non-linear curve, but on semi-log, you should see it as a linear. So for those, because I'm letting you know this now, because I saw this to be a case, and I want to make sure you guys don't not only lose marks for that, but also your future calculations thereafter. So if you have a case that is non-linear, like if your curves appear like squiggly or like move up and down, like sigmoidal, like this, I want you to fix this before the end of today's lab. So that way, like you can feel more confident in your results and in your values. And if you need a calculator, you can use mine as well. Now, when it comes to values that you should see, I said that the range is variable, right? Because after all, you're doing this manually. And I said that you should see a range from 15 to 75. That's a large range, but I'm trying to be generous essentially. Technically, we see it's a range more towards 20 to 35, 20 to 30. And that should be your closest range. If it's a little bit lower than 20, like 19 or 18 minutes, that's okay, or even 17. But I'm letting you know this now so that way you could feel confident with your submission. Folks, another thing is your curves should be so it should be a line of best fit that you're showing for your slopes. It's one that you can show them, like connect all the dots together. That's okay. But when you're doing your line of best fit, that's different, right? Because hypothetically, if you have something like this, where the dots connect like this, you're not going to really get a strong indication of your slope, especially at time points where there should be a generation time present, right? Between x1 and x2. So you want to just bear that in mind. Otherwise, your readings may not be as appropriate. If you've drawn it out already, you just need to highlight to me what your line of best fit is essentially, and thereby determine your slope, and thereby calculate generation time dividing 0.3 by that slope that you have. And considering all those variables to answer Khan's question, or generally speaking, your values will differ. So don't be concerned even in your same practical group that you find someone beside you has 17 minutes as their generation time at 37 degrees for viable counts, whereas the person on your right has it at like 23 minutes, and that shouldn't be a problem. But I want to make sure that you're able to deduce correctly, accurately your generation time values based on the slope and results of your respective graphs. Remember, you have 4 curves. That should suffice. I know we have to get straight ahead, so let me just go straight to the practical. Before taking tubes, Garvin, let's wait just a moment, and everyone as well. I'm going to highlight a few things for today's practical that I want you to bear in mind. Actually, I need everyone to be seated. So, okay. Before you take things, I need to be. It's okay. The reason I'm asking is because this week's practical is a bit different from our past. We only have our supplies are limited to 5 groups, 4 to 5 groups, meaning that we're going to let you separate differently. I noticed this group is a bit more in number than this one. So what we'll do is keep the 3 blocks that you put forward at the top here. But I'm just going to see 3. We may split up into halves. So we'll split up into halves. I know it's not too different, but you guys can work together and be able to once again try to coordinate tasks per exercise. And so I'll talk about those exercises right now. Remember as well, once again, aseptic technique. When you're here in your benches, you want to make sure you reduce any risks of cross-contamination within your bacterium, but also of yourself and additional bacteria that you'll be using today. If you have a look at your red trays, you should see 5 different types of bacterium. If you have, and you'll likely see them, if not more. Oh, you have even more. Perfect. So, and because you'll be using different bacterium, some of which you already are aware of, but the idea, inshallah, is that you're going to be focusing on the following. You have, for exercise 1, 5 different tubes. Hopefully, you have a control that's labeled. If you don't, I'll find one for you guys. But essentially, each group, as I talked about, and I'll have 5 today for at least exercise 1, will essentially separate to do one of the following conditions. I'll have 3 different locations. But the idea is, if we look back, group 1 will be at 10 degrees. 10 degrees is where essentially after you prep up your bacterium with the 10 microliter sterile inoculators, the ones that you find here, group members, please pay attention. Make sure that you just grab the amount of supplies that you need per group, limited amount. But the goal is that you just record the points that I mentioned for your conditions, so that way when you separate into groups, you'll be able to easily. Okay. The point is, the goal for exercise 1 is that you have 5 different temperatures with the 5 different tubes that we'll be using. You'll notice that, by the way, from your manual, there are different classifications of bacterium, right? We've looked into the past of like their morphology, different traits, gram staining, underscore staining, etc. And but in this regard, we're now looking at environmental conditions, as you saw from the first slide. And so, for temperature, you have different regions or ranges in which bacterium are able to optimally grow, right? So that maybe connects us back to our generation time assignment. But the point is, you now are able to explore those changes across the different bacterium. And you'll notice by the names or the suffixes what they could imply. Now, nonetheless, from your results, or sorry, from your red trays, you should be able to see those 5 different tubes, or sorry, 4 different tubes of bacterium. And based on your group, which I'll be isolating you into now, group 1 will be at 10 degrees, placing their inoculated Na broth cultures into 10 degrees, which is the top left, actually the bottom left side of our location bridge. I'll place now. Pretty much very mindful the different conditions, because it's important that you place them correctly. Group 1. How many stage difference? Yeah, I have to make space. So actually, you guys, group 1, to place them where you see these cupboards. I'll have them literally right here. So you'll place these here for 10 degrees. Group 2 will be at 20 degrees. It's assumed to be room temperature, which is we'll have, you can also use the blue trays that are shown there. And then for group 3, which will be at, I believe, 37 or 30 degrees, you'll be actually at the previous incubator, right? So bear in mind, folks, there's a change in temperatures. In this case, because we're dealing at 30 degrees, only place for exercise 1, the tubes that you have, which is technically group 3, here in the top left side. For our place for thermal decline, we'll be using the top left side. So please bear in mind that this is the location. And I will actually need us to split it in half into 4. I'll place their tubes. Let's I'll place it graph. And then about 39 degrees will be where you'll place your tubes. And then 60 degrees, where I just want you guys to be very careful, is here is water drop. You'll see already there's tubes here. But just bear in mind this is hot water, so don't get yourself in too close to that hot water. And if you need help, does that answer any questions, by the way, regarding the separation of groups or your tasks for exercise 1? If you look at your bench tops, you should have, actually, let me highlight this now. So Garden went ahead and took 10 different liquid drops. Please bear in mind, folks, you do not tilt this over. This is not solid agar, because essentially you can have your bacterium and your nutrient broth spill over, and we may not have extra. And that includes your other tubes for the different conditions we have to take. So please bear that in mind. But nonetheless, you'll have 10 different tubes, each group. And the aim is that you'll replicate this twice, because you have 4 different bacteria based on what you see on the red trays labeled there on the screen. And then you'll have a control, which is pretty much without any bacteria. So you can leave it as is to label all tubes accurately on the surface of the tube itself. Include details of the practical, your group letters, right? So I'll place those details now, including the conditions, so the exercise, the bacteria, and the date, right? So this will help you not only for this exercise, but inshallah for other ones. So that's EX1. Let's say today's date is maybe 20. So we're at 20. I said bacteria, and there was something else. That's, okay, we'll say bacterium. So hypothetically, it's E. coli. Right, and just make sure, folks, that you follow the steps correctly. Make sure that you, and actually, yeah, that was the thing. Place your group letter. So you could be group like GRA, for example, like group A for short, or group B, C, D, and then just make sure that you confirm those conditions, so that way you don't mistakenly place the wrong bacterium in the wrong tube. So just make sure as you go forward, you're able to label those tubes accurately. Any questions before I talk about exercise 2 and 3? No questions? Exercises 2 and 3. So exercise 2 will be in the same groups. And then, so essentially, you'll be doing 3 different bacterium plus a control, which was like before. But the goal in this regard is that you'll be looking at differences in pH. So think of acidity, hydrogen or hydromium ions, and essentially the influence that has on certain bacterium in their population growth patterns. So think about that. You'll be actually looking for across the 3 exercises of pH, salinity, and temperature. Next week, inshallah, the results of turbidity qualitatively compare and see the levels of cloudiness on a range of 1 to 5. And then on using the Spec 20, we hope to also, inshallah, look at those results more quantitatively. That revolves around a bit more nuanced stuff. We'll talk about that when needed in week 9. And I forgot to say, congrats, folks. You're like 80% into the practical. Well done. So we'll hopefully get to appreciate this more in due time. Now, regarding a few other things, yes. So you'll be separated into groups as before. Group 1, pH 2, group 2, pH 4, group 3, pH 6, and group 4, pH 8, group 5, pH 10. And so just bear in mind that you keep note of these details and then incubate them. We'll try to place all the tubes back into one single blue rack, and then we'll place them into the 37 degrees. I'm going to go down, talk a bit about osmolarity. This is where it's different because now, as you see, there's only 4 different conditions. And so for the second last exercise 3 and 4, this is where we're actually going to be split up into quadrants. So essentially into quarters, meaning that this is now group 1 here, group 2 here, and then group 3 technically group 4, and then down there group 3. So I would say for now, let's focus on completing exercises 1 and 2 as the groups of 5 or the total of 5 groups. And then exercise 3, pretty much in that regard, you'll only be doing 1 condition. And I know it seems reverse in the lab manual, but this will make it easier so that way we don't have to, I think we have more too. Okay, actually, see, the reason I separated it in this regard was to make it easier, but maybe for exercises 1 and 3, 2, 3, you guys could stay in those groups apart. It's just now when you're following the manual, bear in mind that you're taking 1 of each for salinity. So please make sure you label them accurately. You don't want to essentially mislabel. Make sure that if you have 2 members here, they know, okay, I'm going to put 2, or actually with my right hand, for example, 5% PCL. And then with another region on my hand, on the palm, 2% or 8% or 11%. You really want to make sure you're taking the right conditions because they all, generally speaking, look similar, except the ones that are colored. So just bear those details in mind. Carlos had a question.
Yes, the first exercise, 5 for the second, and 4 for the third.
Technically, yes. So my last practical, we did it the other way because that's why I wanted to know the number of people here. It's a bit different, but essentially that's that kind of 10, 5, and 4. Controls will actually be the extra tubes. Salinity, you won't have a control. So you'll have the 3 different bacterium, some of which, like the bacterium that you already see, may overlap. So when you're doing like your post-lab, you should maybe at least next week be able to categorize specific details of your bacterium. For example, we know E. coli is gram-negative, right? Is the catalase positive or negative? Does anyone know? Negative, right? Or it has a low concentration of catalase as an enzyme. So we can assume it's a low in concentration. And then think about all those other features. It's endospore negative, or if it's capsule negative, etc., or non-encapsulated. So think about those specific details because they'll help you out for your lab exam. And the other point about, like, I didn't talk about this, but the suffixes. So, for example, you see bacillus or B thermophilus, that should give you a hint as to what range it would likely live in terms of its conditions and where it would best or most optimally grow under. Right? So, like, think about thermal, think about cryo. They respectively are like antonyms. Thermal is more like in hotter regions, cryo is more cold, like cryopreservation, which really relates to different types of electron microscopy. But essentially, you'll see the different values or different bacterial species. And my request for you all is please make sure that you're attentive with your labels, with your bacterium, because I want to make sure that you have what you need, but also don't run the risk of wasting or losing any material. I want us to be done exercises 1 to 3. Remember, in those groups of 5, right? With the values that I've talked about. Exercises 1 and 2, specific conditions, right? I talked about temperature. I talked about pH. But salinity, exercise 3, you're going to do all conditions from 2 to 11%. So just bear that in mind. I mean, it brings up the question, what do you use for a control? Technically speaking, it would be 0%, right? So, I mean, let's see. I don't know if we should. Yeah, let's to keep it simple, follow through with the methods in the manual. But nonetheless, I look forward to the best with you guys in our second or our third last practical together. I want us to be done in the next hour or so, and I'm gonna give you guys, I'll put a timer on for an hour and 4 minutes. Any questions, by the way, before we go ahead? Yes, Garba. Okay, so groups are, let me just keep this back on. Groups are 1, 2, 3, 4. So you guys are now split in half. 4 and then 5. So you're a group of 6, including, so group 4 includes now Jacob and Saad, and then now Melina and Alta are going to be on this group, group 6. So you guys will be one group altogether. And let's see how far we can go, folks. Any other questions? No questions?
Our groups divide exercise 3.
Exercise 3, actually, you guys stay as is.
We're not changing.
We won't change it. We won't change it. Are we ready to go? Yes? Are you guys okay with the group orientations? Is that okay? If anyone doesn't feel comfortable, let me know. Are your marks? That's it. Go. If you're done with your generation time assignment, you can actually place it right here. Please make sure you have your name on all pieces of paper if there are more than one. If it's stapled, then it should be okay. But just in case, place it at the back. Apparently, so I think it was, I had one, but it doesn't, I don't know if it works. I don't think it works. Okay. But it's okay. It's okay if you're not. You could manually. That would be a great one. But they have to go in. So, like what I mean is that even when you have them in smaller increments, you want to have them essentially if this was technically like if this was one, not there should be a 10-fold increase. If you did it correctly, then it shouldn't be a problem. I can't help the line here.
As long as it has a name on it.
You're confident?
Yes, I don't comprehend everything.
So let's say these are increments. You have like two. Like, oh no, how did you do it?
Zero.
How did you do it?
I did it, I just counted by like, and then like nine. So I thought it would just be because.
Because what I was also gonna ask, okay, let's see how you results. This one is just going a bit over, but let's see. Okay, I have it. Maybe like the increments,
Yeah,
right?
by my increments, not by me.
Yeah, because technically that's what you want to do. Like this would be, this actually starts at 1, by the way, 0 point. So this is the microphone unit.
These are my.
Okay. So technically you would actually go, let's say 0.01 to 0. So this is 1 inch.
So 0.02, this is 0. Let's see. No, so then I just dropped it according to.
Because they look like even increments.
Yeah.
But technically you're going up like logarithmically. So do you have more, by the way? Is there anything else? Or this is okay, perfect.
What? You doing Barakatuhu?
So 10 tubes, by the way, 5 different conditions, 4 bacteria plus 1 control times 2. Just bear that in mind, folks. Control is just the liquid broth. You just keep it as is. Just make sure you label the tube itself. No.
And I'm sorry for the thousands of emails before I just wanted to, I put a lot.
This is the other one that is like this. That's okay, and then this one sort of see
Yes,
And so you kind of make one.
yes, which is the next one.
I see. Equally distant.
Yes.
Yes, you see, is this the right?
Yeah.
So technically, it's successful. We can do it because you'll notice these two are almost but you're adding in more distant away from you. It may be just a bit. So I would just like but if you are able to adjust it,
So just move it down. Pencil or eraser or anything.
then it can be okay.
It's fine, okay, I'm gonna see if she's for it, and I'm just gonna leave it with you. I'm gonna leave that there. I'm gonna walk away, thank you.
No problem. No. Incubations, actually, yes, great job. Oh, I should show the screen. You can place it right there.
And he said like, because It's a line of estimate. I didn't draw the curve. Is that okay?
That's okay.
I just labeled the points.
That's okay.
Thank you.
No problem. One. If your groups, I'll be on exercise 1, the slide here on the PowerPoint. If your groups are done, please let me know. So that way, if all groups are done, we can move forward in the slides. My recommendation is as you are now you are groups of 5 for 3 exercises. See after the temperatures, if you can even split up, right? Like maybe for exercise 2 and 3, or 1, 2, and then coming back at 3. So that way you guys can complete the tasks efficiently.
Get a drop at the time microbial or whatever it's off.
I'm going to show you guys. Folks, for the exercise 2 are the pH ones. Question. Yes. In these aseptic sterile inoculating loops, your 10 microliter end is the larger circle. If you use the other side, make sure that you bear in mind that you want to use the other one, the larger circle compared to the other one. The other one is 1 microliter. So just bear in mind that you take essentially resuspend your bacterium. Remember, as always, resuspend your bacterium. Make sure that they're turbid, as you would see post incubation, and then essentially with that inoculating loop, following the step-in technique. You don't have to flame the loop itself because it's plastic, but then it's sterile, so you'll just essentially inoculate accordingly your bacteria. Okay.
Well, like, I was picturing it.
Yeah.
Just a reminder, you have for exercises 1 and 2 controls. So please bear in mind for exercise 2, you also have a control, 3 bacterium, and 1 control, multiplied by 2. It's okay if you don't do so. It's okay to label both sides of your of your cube. Sorry, your paper. I'm just gonna be a bit occupied. I forgot to mention this, but bear in mind that I may be a bit busy with a few things. So please don't mind. But sorry, that's please essentially don't worry. You can place them. You can try to manually tilt the tip of your paper so that way they're connected. And that should be okay. Any other questions? It looks good. I just put this on some. I would. Yeah. Yeah, so okay, label containers can be I mean for this case, some racks or trays are enough for like 12 cubes, so it makes sense that you can only use those. But technically, for our upcoming exercises, we're aiming to put all of the cubes in one location because exercise 1, 2, and 3 will all grow, at least for the ones that are 37 degrees for exercise 1, they'll all go in one location. For exercise 1, depending on where you are, for 1, 3, I have this 2 for you guys. So, for that, so give me just a few moments.
Aging at a rapid rate. This is for us. They don't know. They'll go inside.
If you find that bubble seat thin, Carlos, if it's too thin, then redo it. Okay. Actually, for this one, you guys can actually place them directly. Just make sure that you know a number and then your location. All of them are labeled.
Yeah.
And you have
Yes, I'm pretty sure touch the water to
zip code. I would use
Okay.
then. So pen should be. That's good. If they're around the same level, oh, I would place it actually. Hopefully the way I can. Did they get an update how you have to four or exercise one? Oh, this is a reminder. I know I have a reminder and it may seem like you guys are a bit rushed, but you don't have to rush too much. In fact, this is great helping you guys to, balance time with efficiency. So I want to make sure that you guys are, of course, importantly, maximizing your efficiency, making sure you're being sterile and following septic techniques, and not running the risk of any contamination or poor yield. So don't worry. Try to coordinate time. So another good question. After the thermal death time, as a group on it, and as you guys do Bible, prepare your place for speculating, treatment, you guys are going to have a presentation. So as you probably saw from the first slide, you'll be looking at your DB results. So once you're done exercise 3, let me know, and I can take out it's accordingly so you can read them. You may have also saw my previous talk message talking about the results. There may be a bit poor results, but nonetheless, feel free to discuss whatever you can. And if they are concerning, I'll be posting some sample data. Any questions? And then if you have more sterile loops, you have to
The tubes, because we need 4 for the.
Okay, perfect. So, by the way, this is a good point. Make sure that you close your packet essentially with sterile loops, and make sure it's as clean as possible, as sterile as possible. And bear in mind as well that if you're done with specific exercise with specific tools, return them back to the middle tray, all or middle bench, all in one tray. This is a good example. For pHs, you want them to be aligned. So if you're the other one, try to have them in the right location based on your group's pH grade. Like for example, So, please be ready. The one that is on the white. Make sure you label practical number as well. Yes, go ahead.
My group was an extra, can we take a pH right from the middle?
Yes, you can. Just invert it. I actually placed extra ones for you. So, what to do extra for those? Go around. You'll see two trees on that end. Can you place one at 10 degrees, where I was seeing on the inside the right fridge, and one at 30 degrees in the top?
Okay, we're about to put pH. pH in 35.
Oh, yes.
The pH is 37.
Yes, so that would be on. Oh, so okay,
I think so.
we're seeing the other. Now, status update. Are we all done exercise 1? All groups? All groups? Group 1, are we done? Exercise 1? Yes? Okay. Then we're also done. Okay, so we're now moving to exercises 2 and 3. Congrats, folks. But I just want to highlight, try to, in this regard, split up if you haven't done so. So that way there's groups of 2 or 3 or sets of 2 or 3 in each group dealing with a pH or with osmotic pressure. Bear in mind that your group members understand what I'm talking about, so that way they don't run the risk of making an error when you're not aware. For pH, actually, many of you are also done the pH, right? Who's not done the pH? Please let me know now. And it's okay if you're not done, but let me know. As in group 1, are you guys done? pH as well? Okay, let me just see. Yes. Is that you? Are you guys done? Is that the same one or a different one? But I think it was, So in the meantime, see what you can do for the next exercise. I'm going to repeat this.
We're done with concentration.
Any extra sterile loops we have to here? For salinity. Great job. So,
You need to integrate it in
I'm going to give you you could use it. It's about all of them. Yeah, stay on. Yeah. You got to follow number. If you're like 2 goes here, 3 goes here, 2 goes.
Four things.
Sure. This is confirm every group has done exercise 2, correct? Is there any exercise 2? Exercise 3, how are we doing so far? We have another group that we're waiting on. Now, let me go over a bit on regarding your next two slides. If you guys are done,
Once we're done, we'll talk about,
it was hard. Just to confirm, is there a group that hasn't done the salinity test as of yet? Any group that hasn't, because I see extra tubes here. Did you? Group 4? Okay, I'll talk about that. I'm actually taking charge on something for the experiment. Let's hold on for a minute for slide 4. It should be okay. I think it's on the own. It's like really put it here. This one. It could actually have been placed on top of it. And maybe like essentially that could have been already used, but the ones that were above were the. I think this is the must. Folks, did you all get 6 plates for exercise 4? Yes or no? Oh, okay. Okay, I'll give you this one. I believe it's the same. I'm going to just do a few Okay, so it looks like we're all done. Just to confirm, raise your hand if your group is done. Exercise 1, exercise 2, exercise 3. Are you guys all done? Yeah, all done, all done. Todd, are you done? Are you all done? Exercise 1, 2, 3. I'm looking at those. Don't take it wrong that I'm seeing this because I want to also see that you're paying attention. All groups are done. Exercises 1, 2, 3. Okay. So let's just and forgive me if I pointed you out. Now, oh, I forgot one other thing. Before I talk, let me set up things. Once again, folks, there were technically 5 racks of tubes. So unless a group accidentally forgot or maybe used one of the other tubes, please make sure that you actually took the right ones for salinity. So make sure if you've seen your tubes, the ones that are here, they should be labeled correctly, right? I want to make sure that you're not doing the one with temperatures because we had extras for those. But you did it, right? Okay. And whoever grabbed them as well, just please bear that in mind. I'll place a few things. So, sorry, give me a moment. I didn't place them into in the meantime, label your plates based on the final dilution factors, the exercise, group letter, the condition that you're going to be in. The bench beside me right now is the group that will likely be doing more than they'll put up into math. So unlike those groups there, you already didn't. Yeah, that's what you label them, but what is the label of that? 2 or 3? The thing is global, technically still have to split. But then how are they gonna? Let's confirm. Let's play this one. Like you want to make sure everyone the bench is right? So let's confirm with them as well. You guys can coordinate. Then coordinate the edges of this bench, allowing me to that every
It's up to you which group.
If you guys are done, finalize the few others.
Okay.
So let me now go over exercise 4 for you guys. So this is our last exercise today, but it takes a bit of time. So just bear in mind that you now are all paying attention. There are things that I will direct you guys to do, and so just bear in mind the conditions. So for thermal death time, it's going to be like the inverse. And actually, maybe I should ask, what do you guys know based on your lab manual about thermal death time? What are you trying to observe?
The amount of time to go specific, amount of activity on a log.
Yes, yes. And so think about how that compares with the generation time assignment, because you saw over time, essentially, an increase to perhaps the plateau of your bacterium, right? Either being from the microplate reader or from the viable counts. So the idea is, in this regard, you're doing the opposite at different temperatures. And so we're separating you out into those quadrants. Let's pause the timer. Let me see time. I want to know how much time you need for this. You probably have half an hour. Now, before we move further, so you are split into quadrants. Let's see the manual as well, because I want to make sure you guys understand. 4 conditions, based on what you saw, hopefully, from page, I think, 1716, yes, is the following table. Group, so essentially, you have to be very cognizant as a group as to what your conditions are, because some of you are going to get bacterium. For me, think about like the generation time assignment, how you did viable counts in the past, following different incubation time points. In this regard, I have 2 different flasks, one at 50 degrees, one at 65 degrees of E. coli in log phase. And so the idea is that what you need to do is bear in mind your condition and be able to take accordingly 1 mil for your tubes, right? For your starting tube for serial dilution, and I'll show the PowerPoint soon. And then 0.1 mils for your plate, your starting plate. So if we look down, and actually to separate you guys now, group 1 is going to be 50 degrees, group 2 is for 65 degrees, and then it's essentially the same here, except you're doing at time 0 and time 5 minutes, meaning that those that get it from me as soon as possible, like essentially your group members need to be present. And as soon as I actually place in, just like how we did in the past, the E. coli into the flask, the 2 benches or 2 quadrants here have to bear in mind that they have a timer for 5 minutes. Because I'll be also doing things at the same time. And I just want to bear in mind, I likely will be very preoccupied, so I can't really answer questions unless you have them now. Yes. 0 minutes from now. But the temperatures will be 50 degrees on that side for the first half or the back half of the slab. And then the one that's closer to me, this half will be 65 degrees. See, the other thing is that you guys may also need to move spots. So if you guys are cool with that, then that's something you want to consider. Yeah? Okay, okay. And by the way, just to confirm, you guys are okay with the arrangement now. Yeah, perfect. Okay. Once again, actually, let's go now to the PowerPoint. Any other questions? Yes, go ahead. You should have. Maybe other members of your group have 6 plates.
Yes.
So before I move further now, let's have a look at the PowerPoint. I want you to follow through with the guidelines shown here because it will make it easier for understanding. I talked about how essentially I'm starting off with E. coli. They're already inside about 50 and 65 degrees roughly in the water baths. And essentially at 0 minutes, as soon as I place in the E. coli, I or yourself will have to take out 1 ml of the new essentially broth with bacterium and then place them following their dilution into your respective tubes. So have a look at this. You have an original stock, right, at 50 or 65 as shown there. And then you need to have your 5 tubes, 5 saline tubes, which some of you have already gotten from this end here. There's a group maybe that hasn't gotten it, so you guys can grab it when you need to. Just make sure, as always, label your tubes, right? Because you're dealing with different dilutions and you don't want to overtake nor take twice from the same tube, right? So because that's very important to take note of. The serial dilutions, unlike our plaque assay, you're not doing at 0.1 mils in one tube, but all of them go from 1 mil iteration. So you're doing 1 mil from the first tube that I give you, right, or the first tube that you come with following the incubation. Thereafter, you do them in the other set of tubes, and you just make sure that as shown before, and to reiterate this, if you have essentially a original stock, let's say, and we can actually look at the concentration, by the way, many of you should actually consider what the concentration is of the E. coli based on the final volume. But let's say hypothetically, and these values are by the way relative to that original concentration, but essentially because you're taking 1 mil into 10 mils, or 1 mil into 9 mils saline, which would make up 10 mils, then you're getting a dilution of 10 to the minus 1. Because you're taking 1 mil now into of this, 10 to the minus 1 dilution into this tube that will essentially become 10 mils, you'll do essentially a repeat. That's where serial dilutions come in again. From 10 to the minus 1 to 10 to the minus 2 to 10 to the minus 3 to 10 to the minus 4 to 10 to the minus 5, following with 1 mil reiteration. And then essentially you're doing what you've done in the past in the plaque assay. You're taking out 0.1 mils, and the same applies to the stock. So when you're doing it from the stock, you're doing 0.1 mils. Technically, this is 10 to the minus 1 as it shows here, but you want to bear in mind the relative abundance because that dilution is essentially what's going to be replicated here in concentration. So 10 to the minus 1 is here, but you're taking 0.1 out of here. So it's like 10 to the minus 1 times 10 to the minus 1, that's 10 to the minus 2, 10 to the minus 2, 10 to the minus 3, when you divide by 10. And you do the same for the remaining 3 sets of plates. Remember, you have 6 plates. You label them with a practical, your group letters, the exercise for your factors and the bacterial time points and temperature. So bear that in mind. So that way next week, inshallah, you see your results and you get shining into the sun. So that's a bit about my final message for you guys for our exercise for today. Any final questions? Any final questions, thoughts, concerns? Ice? Ice or eyes? Eyes? Find out. You need it? I think mine's- actually, once we go to class- You need ice? Yes. So let's now okay. So let me get started actually, because as you see from the following slides, we also have the microplate reader. So just like the generation time, how you saw the microplate absorbance values, bear in mind that there are going to be other conditions. And they're going to be going down in iteration. So I'm going to ask if someone could actually bring their phone if they've been using it today, because I'm going to have to be doing time points regularly for the microplate reader based on the different conditions that we see here. And the goal is, inshallah, that you want to see this flow, right? Because once again, you're doing it in log paper, or essentially in following the y-axis in log. So just like how I showed you earlier, you don't have to graph it, I'm just giving you a depiction of it. Actually, in your post lab, you should draw it up. You should draw it up. It's not too difficult. Like back in my time, we wouldn't we would actually, I would do graphs without like graphic paper, so I would just do it on line paper. So like it is easier, right? So you guys can do it, but nonetheless, you guys can do the same and then just inverse accordingly. Even if it's a sketch to depict what you would expect to see from the from your results. So we're gonna do this for about half an hour. Okay. Let's see. I have to give myself some time to do this, so because I'm dealing with the microplate reader. So the goal is that you guys okay, let me think. Sorry. Okay. So essentially, we'll get ready in a minute or so. I'm gonna start prepping up the microplate reader because I have to do these incubations. But if anyone has a timer that I could use, if you're comfortable, because I'm dealing with two different groups at the same time, so it's a bit difficult. Any time point work for me? Top watch. By the way, if you guys are comfortable, because technically it's on a bench top, so if you guys are worried about bacterial. Because I do technically, but I don't want you guys to. I appreciate it. I appreciate it. I can try. Thank you so much. No, because I don't, and also the thing is, because you guys are a group of five. No. In fact, I'll take everyone's phone. With the phone. No, you want to? That's okay. Oh. Okay, you know what? Please interrupt. I'm worried. I don't want anything happening. So, okay, let me just get started. Pipette tips. Is that your question about pipettes? No problem. So, in the meantime, you guys can grab pipettes if you want. See, I didn't want to interrupt you guys. That's why I ignored. So try to do them in the meantime. Try to complete exercise 4, because you guys will have time to complete.
Can I use that?
So, well, I would appreciate if your groups are paying attention for timing. Focus on exercise 4, because that's more important right now. So once again, the one that's just technically, you're not just technically, you do that 65 minutes in the sorry, because I'm at here. Because there's something I have to do in the meantime. Yeah, folks, don't focus on the police right now. Focus on exercise 4. Focus on exercise 4 so that way you guys are able to all be able to complete the lab with efficiency. That's more important for priority right now.
I mean, please.
This is the one that Please come here. Yeah, so you're gonna have your pipette with your point. You need to put it for 100 microliters. Put a pipette for 100 microliters for your tube, for your plate.
Still quality.
Yeah. Each group should come with their plate and their tube.
As the -1, so plate value for first time.
That's for 100 microliters, so you need.
Yeah, that's fine.
Are you ready? For 100 microliters. I'll do it. I'll do it. I'll do it. So you guys, if you want to, that's okay. You want to put it on Folks, I need you to come with your plate and your tube. You need to have a pipette at 100 microliters, please. It's okay if the thing is this is a bit sensitive, so I just like you guys it's a bit try not to take it. The sooner that you guys can get it ready for me, the better.
That was another exercise. We gotta get that one.
Can I borrow if you want okay. Just for five. Actually, it should be okay. When you guys need it, let me know.
Okay.
If that's okay. Because the other groups will be using it very shortly as well. Is that okay, Carlo?
All 7.
That's okay. Ready?
But during the upside down.
This really mine. This week. 59. Yeah, that's right. Folks, are you all okay? Everything's okay? We'll wait another 20 seconds, because it was outside of it. Yeah, of course, take your time. So yeah, that was the problem, because we haven't heard you usually.
Yeah.
So you restarted. Yeah, that's right. So your group should not be. Okay, you need a tube. You need a tube and your plate. You're starting to do that. Did you? All tubes are contaminated? Yeah. That's good. Okay. So, in the meantime, please, can you just please, now,
Number 12. This is good. This is.
So now you can come in with
Okay, 15, not I'm going.
50 meters of credit. There's the so Dr. Brent also I could do it this way to separately see how it goes. It's already cut on. You ready to go? I think they passed. Are you comfortable with it being left here? It's okay. It's okay. You can say it. But always at this time. 12:05 needs to bear in mind the timing in the next minute. I'll give you a minute. Give me a minute. Fine, garbage. You say you. Yeah, we did. There. Once we're done, we'll talk. Or if not, then after the poll, I'm going to be a bit time here, so just be a bit cognizant. I may not be able to answer questions the next 10 minutes. And if you have your questions ready, I could try to improvise the coding. Think about what could have happened. Harvey, what's the time? Please come with your plate and your tube for 0.1 mils of your pipette.
I'll take the other ones for fun. Let's see what.
Yeah, that's what I'm looking at. Sorry, what was your question? Wait, you are playing around? Oh, I haven't seen. Okay, you can do this. Okay, you guys do this? You can.
Okay.
What was the question?
Okay, never mind.
It is actually related.
Very important.
I appreciate it. Forgive me, folks, if I'm like very tense at this time. I want to make sure that like I'll try to do my best to see if you guys can like to help you guys get the best grade accordingly for this exercise. So just try to bear in mind that I need your support accordingly. So in the meantime, if your plates are done for spread plating, you can take a photo, you can invert them. Both, let's pay attention. Also, safety concern, please make sure that your lab coats are buttoned up, right? Because you don't want any bacterium coming directly into your clothing. Another important reminder, please. Folks, are we paying attention? So this is what we're going to do. When you're done exercise 4, make sure that you have your plates inverted and then tape them around for the exercise with the label that I talked about before into the top left incubator. You need to bear that in mind. And then thus, it's different from our previous exercises together. Everything okay?
So just maybe a labeling.
If you guys can have your plates, there may be extra plates in the fridge for exercise 1. Please make sure that you grab the plates also for exercise 1. Okay, I'll direct you guys. Back for the as you're going to be going forward with the presentation, so make sure that you have your lecture. I think prior to that. I just want to highlight that, the wrong way. You can bring, so we have exercise 1 plates here. I have them provided here. For the next few minutes, you guys can continue preparing, but we're going to start at 30. So just bear in mind, in your groups of 4 from before, try to highlight in the next 4 minutes for each group your results, your conclusions and implications. I likely won't be able to ask questions, but please try to stick with the timing of answering what we saw earlier in the board. Provide your intro. Everyone, are we paying attention? Are we paying attention, folks? So I appreciate it. Thank you for letting me know. So I just want to highlight, sometimes when things are fast-paced, I can't answer questions, and that's why I preferred actually to do the plate rearrangement before, so that way we could complete the TDT exercise earlier. Where we are right now, you have a few minutes to just finally go over the intro results and discussion. Try to cap your, and that's based on your second exercise. You can talk about a bit briefly of your first, because I know some of you just got to see your plates. But prioritize the discussion with all group members conversing or presenting their data. They could present it at their benches as well. And try to stick with timing. Any final questions before you begin? No? Yes, sir. Saline tubes will be placed down at that end where the other tubes are located. Try to have them all arranged in a single tray, so that way it'll be easier. Yes. In your groups you're presenting. Yeah. But yes, you're presenting as groups. Yes, in front of, like, so you could stay at your benches, that's okay, but you'll be presenting to the class your results. Just bear in mind, folks, that you're all taking turns in the presentation. It could be a minute or so per person, talking about the intro, the results, the discussion, and it's very simple and brief. I just want you to highlight though the implications.
One set of data at one time is not the role of the experiment, or is it the role of the laboratory? You do this experiment as a class, not as a group. Group is responsible for one time for it. But in order to interpret the same, you have to see evidence. Just like each that is assembled up. So you should be assembling the data all in one big data set and let them look at one big data set to make their.
So that's correct. And the reason though I was telling them to do the presentations as another way is to provide it easier.
Absolutely no sense. That was clear. And it's clear what's up with the top. You have them assembled and give them a chance to look at the data the way it's supposed to be looked at. And so everyone take your data, put it in this bench here in order, label the times, and put it together as one big data set.
So don't worry, what you guys can do is just sort them out, and then we'll likely spend a few minutes. We'll do them as a group all together, so that way it'll maybe make things more easier for trends and analysis. And That was a great question, Hajra, about like, okay, what do we do for presentations? I said group because I wanted to make it more customized for what you guys did, so that way everyone could start taking notes accordingly. But as a group, we'll come all together and we'll just describe what we have and hopefully finish even sooner.
Okay. Why? This is
Everybody get a lot of bacteria on the mask side. That's one of the and if you have mass side and inoculate unexpected blood bacteria here and there. So does that tell you about that? Yeah, they probably run the liver. So they probably didn't operate. So there's probably operations. What you want to do is make sure you see the line and then look for a trend. So if you look at like whole life first, right? So you have your times. What's your first point? Right. So you don't expect to see anything happen. Right? Okay. So would you conclude that in all conditions that's what you would see? Keep in mind that you all did a set rates. So you all didn't equally low the amount of bacteria. So there's variability there. So you have to keep that in mind as you your data of your trend. So if you look at E. coli, what is your conclusion about E. coli? In terms of when kills. So I don't know if it's complete at 5 days, right? Because remember, you allow things to grow after you expose, so if anything that's not showing growth on the A side is dead. Okay. What about the staff? Now look at this 24 hours versus the 72-hour culture. Look at this first. Okay, so we're probably not problem. Pay attention. Here is your A side. Here is your. If you look carefully, you start with the C side. So even at 10, it's a little bit here. But at 10 minutes, it's over here. It is like and 7. This you might say, well, it's not filling up here. That's it's not enough here. The same thing happens here. You're starting with some killing cases, and you're complicated by the fact that your inoculations are not. You're seeing a trend that death occurs faster in the 24-hour culture. Based on what you know about endospores and the release of endospores, what do endospores get released? Did the endospore stay? Did the endospore stay? Did you use the same age culture from what versus the grand state? What did you have to do? You used the older culture. Because the endospore started to form as culture ages. So as the culture ages, the cell subcultures will form more mature endospores that are released from the cell. So what this is showing you is, yes, there's less killing of the 24-hour culture, which had less maturity. It started before the next 7 days. There does not seem, obviously there are variabilities, and you need to point out that there is particular history. You're supposed to get along. This is not what you were supposed to see. This is what you're supposed to see. So when you did your plating, you have to follow the directions. You pick the box off, go like this, go back in, turn box off, where you go on back here. That was important for this experiment. But you still can interpret the data that sits here because it's showing you the trend. You want to take this data and then walk over and look at that data to see more clear. So based on this and then that, make conclusions and talk as a group about the entire data. And between all of you talking, you will get a real understanding of what the point of the lab is and what the data is about. All I have you do is this group, I have to present this, what piece of information you like meeting by this kind of data together. This is not an experiment. This is an experiment. This is the experiment. Okay? So I would suggest you also put this up there, and then all of you do your presentations and talk about trends that they see. Okay?
So yes, you guys could take photos. We'll be using them at good times. And what I do want to highlight though is that based on what you mentioned, yes, you see a trend. But either way, you should be able to know what differences you find out. Because that will give you a better idea based on what you said, not only the performance of the population, what the trends are across culture of bacteria and so, really, you have different ways of interpreting the data. You could do it across time. You could consider comparing across cultures. And that's really the theme that you want to highlight for the report. And my goal is that we'll take a photo, we'll be able to show this profile for this for the exam. That's the one. So just bear that in mind. Your final assignment for the lab. And I know you guys maybe haven't considered if you need support, I'm happy to you guys. But just try to understand the data that you've already made, and you should be able to get the data as we said. So actually, let me just go forward in a moment. And just to confirm, everyone, does anyone have further questions from what's shown here as well? Let's see if I can show. Oh, okay. Okay, so let me just grab some information. Okay. Oh, yeah. Folks, let's just pay attention for a moment. I just want to highlight as well, you have your antimicrobial susceptibility test, right? So let's actually just slow down a bit. Some of us, like myself, are fasting, and I just want you to take a breather for a moment, right? And this is something I want you guys to be a bit aware of. You guys have unique personality, unique traits. Like bacteria, right? They have different traits inside, etc. And I just want to highlight that, I didn't say to our group, but I want you to think positively from our interactions, our exercises, especially with me, because I don't want to at any time make anyone feel like they regret taking this course, at least from, and that's my responsibility. And with that in mind, yes, there are things that change up and maybe unexpected, but I hope overall that inshallah you guys will have a blessed time actually in this practical, and maybe considering the implications of your results in medicine, in laboratory research, in industry, etc. So just bear those details in mind. So we're going to go forward now, and we're going to actually look at the results. Once again, as you could see from what we talked about earlier, you have a trend, right? Across time points, across species. And I actually want you to understand the concepts that we talked about before in two ways. Before I move further, the antimicrobial susceptibility test plates are here from last week. So recall that what we did was we actually took some zones of inhibition for your experimental conditions, essentially one drop spreads across these additional MH plates for subculture. The goal is to actually tell, based on the antibiotics, if they show bactericidal effects or bacteriostatic effects. Bactericidal, i.e. that what you would see in those sections is nothing, whereas bacteriostatic would showcase the presence of those bacteria, meaning that they are still present, but they're no longer able to replicate further outside the conditions that we have here. So we have a few groups that maybe should look at their results. You could grab them, and then I'll answer follow-up questions in due time. I do want to also highlight because actually, maybe all groups can be combined. So, I'll let you look at the results for 5 minutes, and then we'll go through briefly the remainder of the practical. And by that time, I want you to all be able to come, and we'll briefly talk about the results. I'll make it easier for discussion, easier for us to. I know some of you as well may have a biochem quiz coming up, so pretty much take what I've said thus far as key points. Your intro, as we've done from the past, your intro, results, discussion, about in detail what you've observed, be technical with the terms that we discussed, the palettes for a little picture of scientific name for bacteria, and other things. So the issues we have. So, any further questions? So, let's see. Okay, let's talk about this now. A few important things. Bear in mind that your kids, that for your post lab. Okay, actually, you guys can come now. I'll talk about it. And then performing the process, if you look on that side, but this is the zero, the very good. And so when you're taking the result, describe then also you want to talk about the so you see across UV exposure, some places across cultures appear to show no or very limited exposure. And then that increases it. So we look at the all one. A is essentially the one that's sorry, the one that's unmasked. B is the one that's masked. Picking that okay, the one that A should be the right exposure. Technically, B is certain of the exposure. So here that we provide the reflections. And the goal is there after we compare. Let's have a look at the results we provide. Which group was so maybe I'll have you guys engage as a quadrant for 3, 4. So let's look at the for example. Group 1. I want to look at show. This is the group 1. You see that line on the main side is slowly side and speed. So basically, you do see a drop, right? Yeah, so there's 2 things that you want to consider. One is the trend, right, across time, and another trend across bacteria. Because certain bacteria will have different reactions to exposure. Some will take longer, as shown by 24 hours, and others will take slower times, or more quicker, take quick response to exposure. A lot of time. When you compare 24 hours to 7 days, you'll notice also some differences, and likely this will indicate the role, right? You know, what's the role of this? So think about that with the other functions as well, right? So if you want to extend this, then it would be subculture, to be able to hopefully appreciate different sensitivities of all. So the idea is that you have trends across bacteria, trends across time, and you want to be able to describe both and provide. Thereafter, you want to talk about the question. You summarize the discussion result, summary, across time and bacteria. In your discussion, you need to talk about the summary possible. See what the trends are. And that's why I actually appreciate talking as what you guys that way you know maybe my group didn't consist of full one or full one iteration one, but maybe they did it in unique fashion. So just bear in mind like you see some trends to discuss those details in particular and see what that's what I would prefer to actually have you guys do what you've done, so that way you know that's what it should be.
We discuss our specifically and
That's okay. Try to provide limitations like I would say at least two of them, because there is one that's that you can see here that there's other than one. Okay? And even cross contamination. This and I don't want to I prefer not to people's but think about what is the quality system of our team. How do we have the breakdown here. I normally very considerate, right? So wise securing and determination. Even just to like make sure that you all done correctly. So just bear that in mind. Oh yes, we talked about for your intro about UV, right? think about school, think about now the other thing is I wanted us to look at the other results, right? So how about this? Look at your results as a group, and I want you to tell me what you see the plates that like. If you just look at your plates if you have them already. Just tell me what you see. You see there being greater, actually, you see more growth in plates with light exposure as a hair, or plates with I don't know. Okay, actually, you guys can now get to back to your seats, and then we'll briefly talk about it. If you guys have to go, I understand. All the best for current tests, your quizzes, etc. Follow the outline. Yeah. But try to provide benefit. If you've taken photos, yes, but I want you to, okay, before you do so, and remember, what was the purposes of plates 6 and 7? We know those are controls, right? Right? They didn't have exposure to UV. So if you don't see results, and think about those results and what you would have expected for your FLR for exercise 1. If you see trends that are anomalies, you need to describe them. Folks, please make sure that you describe anomalies as well with limitations from the group project, pretty much being able to delineate the different trends that you see. If you have to go for the for a quiz, whoa. Sorry, I'm doing water still, so I'm worried about it overheating. But nonetheless, let's see. One question. One question. Okay. Questions from this side? I know some of you have to go. My last bit for you guys. Okay. I talked about this before. Please make sure that you look based on the slides and also my corpus announcements for further details, understanding the differences between repair mechanisms, determining bacterial statics versus cidal effects of antibiotics. Also know the modes of action if you can. But the other point, yes. For your post-lab, exactly. Not for the FLR. So for the FLR, focus on the results you see there. I'll also try to post a reminder for you to be able to understand what you need, including the TDT absorbance values. There is another point. For slide 7. So not necessarily, no. Reason as well is that these are finite time points and the UV exposure is different. So that's why I want you guys to think about the different methods you guys did. You know what you did in terms of, okay, did I mask, unmask? Did I tilt or turn over my plate accordingly? Did I cover with a lid? And they have important functions, right? But you want to be able to describe those details with me clearly for your FLRs. For the last minute, before you guys leave, I just want to also highlight, so we talked about generation time. Yes, okay. Make sure that you are able to understand your next steps. For the FLR, you have an intro, you have results, you have your discussion. Describe what you see or what I've mentioned thus far. For your lights, and this is the last point before you guys can leave, bear in mind that you are following UV radiation mechanisms from across your conditions. And from the results, if you had your lid off, shouldn't you expect a direct UV exposure? It would be the case, right? So if you didn't, it may have been that you weren't either it was left on or it was inverted. And I know I talked about that before, and I was concerned about your safety, which is why I didn't want any bacterium and aerosols or anything being in your direct exposure. But think about that and light versus dark mechanisms. Dark generally has more advantages for specific bacteria. So to explore and determine whether E. coli uses or performs more optimally in light repair or dark repair, and discriminate those two conditions for your post lab. FLR focuses on the lethal effect, or pretty much what I've already described with you. So with that, hand off. Any questions, folks, before you officially leave? Yeah, I know it's because you're both shown on this slide, but essentially describe in your post lab your analysis from the damage and repair. Take photos if you haven't done so, and you can show them off to me or show them in your lab notebooks. For your FLR, take photos of the one that you saw right there from the class. Take care, everyone. If you have any questions, let me know. Take care, everyone. I apologize if you guys had an FLR, sorry, a lab quiz coming up. All the best. I hope you guys ace it. All of you. No, I'm not lying, by the way. If anyone said that. Sorry? Yes. By the way, please make sure you brought back your generation time assignment, right? There may be some that may be missing. So please, if you haven't given it to me, you need to do so. Yes. Okay. This is the data. The FLR? No, it's another class. Biochemistry with Dr. Brunt, I think. Or Dr. Mock. We don't have a quiz. It was another group, that's why. Okay, so let me just make sure everything is clear. Okay, I'll have a look and just. Everyone in the lab, right? Please make sure that you brought it here so that way I could take it accordingly for marking. Actually, they left it here. You left it here? That's good. All the best. So if you guys are wondering about your unknown assignment, I'll talk about it as a group maybe. So that way you don't actually I think it's maybe better separate. I want to give you guys your time. So let's have a look. So this is the filter. BT is the actually this. Now the thing is when you do this, it should be down that. The slides are also close by. So you can, if you wanted to have a look, you can. The issue is that they want me to leave as soon as possible. So I don't mind if you guys want to look at your results, your assignment. You can have a look at them there, and you can come by even afterwards as well. If they ask, just let them know I needed to have a look at my assignment accordingly. But if you have questions, and you let me know, and you guys can wait accordingly. I'll try to answer them. No problem. Take care. Myself. Sorry, I want to highlight that. You want to separate them, right? So you see 435. That was the one that pretty much from the other ones wasn't fully delineated. So you want to be able to separate all of each other, i.e., from the ones that are caucus, which ones can you separate following additional tests or staining techniques? So that way you would be able to know, okay, this is indeed Micrococcus aureus, and this is indeed Micrococcus nucleus from the very end. That's Does that make sense?
Yeah.
Okay. So then the next one is the quality of the snooze. So the ground was okay. This was actually pretty good. At the end of the score, I think maybe was lacking, and you can probably keep the slide itself. I normally scale a bit, so but nonetheless, I would say that pretty much reflects a few other things. But
I hope that. But I think the other material test was like a because in the two.
who? So what is yeah, okay. So I understand. So technically then that should be okay. Sorry, I'm just worried they want me to leave. But I hope I answered. Yeah. You can keep it with me. And if you see back your slides and for whatever reason there's a mistake or even from our previous assessments, just let me know. With that, I hope you enjoy the rest of the week. No problem. You ready for what? So you want them to so I would okay. What you know is read work and from your you know the reason is because what you can do is leave it on down the bottom and then I'll see if you hopefully collect it after the CDC. If you can try to do it if you have the time with your eyes, that would be good. Do you know when you're done? Like when do you think you'll be done? Yeah. Like because okay, what do you mean by the periodic test?
Those points and pretty slow. I don't like God from
Yes, you don't have to directly connect them, but it should be a line of best fit across the back.
Yeah, that's right.
Okay. So try to redo it and then tell me if you have to update a few things here. Do that and send it to me as soon as perfect. Nope. Honestly, not those ones.
From the other exercise.
Have you already recorded the observation? Okay. Okay. So, um, everything. So you did this correctly. Deform. So I think E for E for program training slide. It was the quality. So it may be treated first with like and like hinges, essentially like ripple, likely, and that's no problem. Have an amazing week, you guys. I hope you enjoy microbiome. I think you do because relative to microbes, a little bit is quite very little. So, all the best. I hope you guys enjoy the practical. Forgive me or Dr. Brunt for shortcomings, but I hope that you guys enjoy it. Try to. Like in life, you have to have patience, and at the end of the day, people will rise beyond the stars. So that's the goal for me. 7, all the best though. Like don't get too distraught. Reply with peace. Take care. 70%. I was reflecting about the slide and research. There's something else I'm missing. If you guys have questions as well, I can try to answer them a bit outside. That's okay. I don't mind if you guys just bear in mind they may ask that you leave. So try to I'll try to answer them a bit outside maybe for those that have like questions about their assignments. For honest, yes, if you want to keep them, you can. If you want to dispose them, it will go to the audit phase. Okay. Take care, Carlos. Everything okay? Sorry, yeah. So, there was one other thing. Okay. I realized I have to pack up because I don't want to also make it inconvenient for them, but I'll answer if you want. Any other questions?
No, this is something I thought earlier. I'm gonna have to take a late part on the, generation time. So mind if I email it to you and I just took it?
You have it here?
No,
Okay. When do you plan to do it?
I'm planning, I'm hopefully getting it to you today.
Try to do that as soon as you can, and essentially the sooner the better. And I likely, because it's the same day, I likely take out. I'll try not to. That's like I want you to be able to do it as soon as possible. Because I understand different people have. That's why I want to make sure.